With that, I'll turn to the Armitage, and Brian touched on the history of the series at the start of the day. But the lecture series started in 2003 and was established to honour Peter Armitage, an outstanding medical statistician who we're very pleased is joining us virtually today. So Peter's online. So thank you, Peter, for joining. Peter's work is recognised really widely and has a truly profound influence on defining the field of biostatistics, with contributions from stochastic models to cancer process to sequential trials methodology, and I think exemplifies what the BSU has tried to do in marrying methodological rigor with understanding of an applied problem and going from that to real medical and public health impacts. And I think you also saw good examples of that earlier in the day from Danny and the other speakers. We'll also see good examples of that from Nick, I'm sure. So as many of you know, Nick is Professor of Biostats and Epidemiology at the London School of Hygiene and Tropical Medicine. Previously, he was a professor at Berkeley for a long time. I won't give the dates, and also vice provost there. And Nick again has made really wide-ranging contributions. In fact, I was just thinking, I think the first of Nick's papers that I read was with Robinson and Jewell, 1991, my notes say, on covariate adjustment in logistic regression, which is still worth reading. It's a very nice combination of rigor and intuition. Since that, Nick's done all kinds of things, but he's best known for work on statistical issues associated with infectious disease studies, including HIV, dengue fever, Ebola, SARS, flu, COVID-19, of course, and we'll hear about some of that work later. Nick's written over 200 articles, a couple of books, and that work has been widely recognised. So I have a long list of prizes here, and I'll pick a couple out just to embarrass him. So the 2012 Zealand Leadership Award from Harvard, the 2005 Snedeker Award, the Distinguished Teaching Award from the University of California, and one I particularly like is one from the Director's Award from the Federal Emergency Management Agency for extraordinary leadership and vision in implementing strategies that enhance the disaster resistance of the University of California, Berkeley. So just showing the range of things that statisticians can do. I will also mention the latest of these, or the latest of which I'm aware, unless you've been given something else recently, is the Mantle Award from the American Statistical Association. Again, contributions to the development and application of statistical science to problems and issues in epidemiology, and particularly noting the recent dengue trial in Indonesia that Nick was pivotal to. And with that, I will shut up and we'll move to the main event of the day. So over to Nick for the 19th Armitage lecture.
Thank you very much, John, for an overly kind introduction. He didn't tell you all the disasters that happened in my life, so it was a very selection bias description of my history and statistics. I would like to start by reminiscing just very briefly about my meeting of Peter Armitage. I can see him online here, and I'm guessing that he won't remember the first of those meetings, but I wanted to just briefly mention them to illustrate the influence of people like Peter on the next generation of statisticians. I happened to meet him for the first time at the London School of Hygiene and Tropical Medicine in 1976, I think. It could have been 75. I was a final year PhD student in pure mathematics, and I had decided that I wanted to do something useful with the rest of my life and somehow had found medical statistics. I knew nothing about it. And somebody sent me in the direction of Peter Armitage. I imagine it was David Finney, who was professor of statistics at Edinburgh, where I was a graduate student. And I hitchhiked down from Edinburgh. I had no money, of course. I was very poor. And believe it or not, Peter, of course, was the head of, I think, medical statistics probably at the LSHTM then. And he made the time, I think an hour, to spend with this unknown kid from Scotland who barely knew a word about what he was talking about. And Peter spent that hour and guided. I was going to the United States for a couple of years, and he made several very cogent suggestions. I ultimately went to Berkeley for a year and Stanford for a year. Peter himself had been at Berkeley before that time. And he probably won't remember that conversation, but he was a very senior person who took the time to talk to someone who knew nothing. Roll forward 15 years, and John mentioned a paper, the only paper he mentioned in his introduction on logistic regression, and I visited Oxford then. Peter was now the head of statistics at Oxford, and I visited as a young faculty member at Berkeley at that point. And our conversations over that 3 or 4 month period were quite influential in that paper. And I'm sure he's mentioned in the introduction as being important. So I'm truly privileged to be here and asked to be here to give this lecture. But I'm most excited because it gives me a chance to look Peter in the eye and say thank you for both of those contributions to my career. And just to say that his attitude to young people and people trying to learn the field heavily influenced my own personal approach to young people now who are interested in statistics. So thank you, Peter. Okay, now I'm going to move on. Normally in the last 10, 20 or 30 years when I've given talks, since my career was very heavily influenced by HIV, I've always sort of taken the opportunity to make a plea to the statisticians in the audience, please pay attention to infectious diseases. Everyone wants to do cancer and cardiovascular, but infectious diseases are really important, and there's lots of need for statisticians. And that whole motivation seems to have disappeared in the last 2 years for some reason. As the 4 previous talks have indicated, there's a lot of really great statisticians now thinking deeply about that. And so this is a little bit of a description of more recent thinking. It was not motivated by COVID-19. This work was really initiated prior to COVID-19, but inevitably COVID-19 will appear briefly. I'll try and keep it as brief as possible to keep things broad. And I also am not going to go into great technical detail. And you can ask me after the talk about technical detail, and I'll probably tell you to talk to me tomorrow. But I'm going to try and keep it broad and try and touch on the parts of this story that I'm going to tell that really do provide significant opportunities for statisticians to have an impact in the science of what's going on. And it's not just a narrow, it's not just a modeling infectious disease dynamics, it's not just vaccine trials. All of this is just replete with the need for informed statistical advice. So I'm going to start the story with talking very briefly about seasonal influenza vaccine. This is a medical statistics audience. I'm sure you've already all had your seasonal flu vaccine for this year, or made the appointment at least. I got mine last Friday. So we get these every year, or we have the opportunity to get them every year. Of course, originally, vaccines are approved for regulatory reasons on the basis of usually 1 or more large-scale randomized efficacy and safety trials. And that was, of course, true for the COVID-19 vaccines also. So these are very large trials. You may have paid attention to them with a little more interest when the COVID vaccines were developed. But influenza vaccines are modified every year, just as we're getting used to boosters and things changing with COVID-19. That's been going on for a long time with influenza because we know the strains differ. Part of the influenza vaccine industry is trying to guess what strains are going to be prevalent in preparation of a vaccine. So these are modified each year to respond to this changing landscape. And the question is, well, if you listen to the BBC, I'm trying to be British-centric, but I'll probably be US-centric at some point. But if you listen to the BBC, at some point in the next few weeks or month, some bright spark will get up and say, this year's influenza vaccine is 60% effective against the strains we're seeing. Or a few years ago, it was 20% effective. Not bad, actually. 20% effective is still pretty reasonable to take. But you hear that information and you might stop for a second and think, how do they know that? So they don't, how do they know that number for this year's influenza vaccine? Because they're not doing it on the basis of a new large trial. There isn't, for many reasons, partly just financial and time, there isn't time to do randomized trials every time you modify a vaccine. The new booster, the new bivalent booster, which I got the week before last, so my arm is still multiple vaccines going on in here, that was not approved in the UK on the basis of a large randomized trial. Unlike the initial vaccine that was directed at the wild type COVID. So they're not possible to do that. And as I said, this is the same story as being told now in the context of COVID-19. So there are not new big trials going on. And with COVID, we have different vaccines and boosters and how long it's been and so on that may, as Danny's talk influence, may influence the level of your immunity, either to infection or onset of disease. And we'd kind of like to know, well, what's the performance of the Moderna vaccine versus the Pfizer vaccine versus other vaccines? What the impact of boosters? How much does that influence the efficacy of a vaccine? Now there's a new variant every so often. How do the vaccines that were designed in the past work for the new variants? Is one vaccine better against the new variant than another? So there's a lot of natural questions that are important to answer, but we don't have big trials to answer all of these. I'm not arguing that for some of these questions, it might not be a bad idea to try randomization, but it's not done a lot, and it's not done for influenza from year to year. And so here's some of the questions, for example. And let me point out that even though I'm not arguing against using randomization when you really can, and in fact, it might have been useful for head-to-head comparison of vaccines early on in COVID-19 to continue randomizing. But some of these questions can't really deal with randomization. In particular, you can't randomize variants to different individuals. So if you want to know, is the Moderna vaccine, how is its efficacy compare for Omicron to Delta to Alpha and so on? That can't be answered by a randomized trial because you can't randomize the variant. And if you think about it a little bit, when you don't have randomization, comparison between variants of that kind is probably, fortunately, less subject to bias than comparison of different vaccines. Because people took different vaccines, these were selected for a whole variety of quite complicated reasons, not the least age, where older people got certain vaccines quicker than other people. So the 2 points I'm trying to make is you can't always do randomized trials, and so you're going to have to depend on observational studies, and some of these observational studies are tricky. They're subject to certain biases. And so the question that you might ask, I know there are experts in the audience who know the answer, so no spoilers, please. But you might, how would you design and analyze a study, given you can't resort to our favorite randomized trial? So how would you do this for the influenza vaccine to give those numbers so that the BBC can tell the public, this is the efficacy of the current seasonal vaccine? So I'm going to stop there just for a minute, let you ponder that, those of you who don't know the answer. I'll just say a little bit briefly about some ways you might have thought of doing this. And early on when I was trying to think about these questions for the first sort of vaccines, I became aware of this this EVES database, which was essentially in Scotland. They took advantage of essentially a national registration of all the vaccinations, and they linked it to a comprehensive database on all COVID-19 virus testing, and were able to therefore infer from that the performance of different vaccination strategies in different age groups and so on. So that's fantastic. Good old Scotland. And if you can't tell from my accent, I'm really from Scotland, not from the United States. So that's terrific. So there are other options potentially available to you in the presence of well-designed, comprehensive national database systems. I'll come back to that at the end. That's really important because you can exploit it immediately. Now, here I'm going US-centric because this is where I was at this point. Imagine trying to do that in the US. Well, I did try to imagine doing it in the US. It's completely impossible. It's completely impossible even within most individual states. As you know, the United States has basically no national health system. So there's no linkage from state to state on health records. But even within a state, we've got a mixture of public and private hospitals, and the private hospitals don't share data with the public because they're in a competitive situation, and there's just no availability of this kind of thing. So the absence of those data really crippled the ability of the people in the United States to answer these questions quickly in a way that at least in some parts of the world who had invested in healthcare system, database systems could. So it's really not possible, what's not possible in the United States. Okay, now I'm going to leave that story just sitting. Well, how would we do it if we don't have the kind of databases that would allow the ability to answer those kind of efficacy questions? And I'm going to change to a different story because I said, actually, there's at least two strains to this story. So I'm now going to talk about something completely different. Still an infectious disease, still a viral disease, which is dengue fever. And I don't know how much of you know much about dengue. It's probably, along with malaria, it's the most important mosquito-borne viral disease. As you all know, the greatest enemy of the human race is the mosquito. It has killed more humans than any other agent in the history of the world by a long shot. And dengue is particularly worrying. It's the fastest growing infectious disease right now in the world. It's gone up. This figure was probably from 5 years or so ago, 30-fold increase over the last 50 years. So it's exploding. There's now probably about half the world's population that are at risk of getting dengue in any given year. And that number is growing every year. And as you can see, close to 400 million infections a year of dengue. And dengue isn't, of course, always lethal, but on reinfection with dengue, I won't get into this story, there are 4 different dengue viruses. The infection of a naive person to the first usually leads to flu-like conditions, somewhat debilitating. It's called the bone break fever, which gives you a sense of the pain that people feel, the aching, but it doesn't usually kill you. But because of cross-immunity reaction, if you then get infected later on in your life by another of the 4 strains, different from the one you now have immunity, it can lead to dengue hemorrhagic fever, which is often lethal. It's definitely a condition that will land you in hospital. And so a fair number of deaths every year. It's transmitted by more than one species of mosquito, but primarily by this one, Aedes aegypti. Aedes aegypti is a little different from the mosquitoes you experience in Cambridge and in most of the Northern Hemisphere. The Northern Hemisphere mosquitoes, largely the ones that buzz around you at night, that whiny sound in the middle of the night, you're trying to sleep and you hear that, so I have to switch the lights on and find a magazine to swat it. And they usually come out at dusk if you're having a barbecue. Not a great idea if you're in mosquito country, because the mosquitoes all come and they start biting you. Aedes aegypti doesn't feed that way. It tends to feed in the middle of the day, so bed nets are less valuable because bed nets protect you at night. But if the mosquito's biting you at lunchtime, you can't walk around with a bed net around you. And they attack from behind, and they don't make that noise quite so much. So it's actually a little harder to detect when you've got an Aedes aegypti near you. So here's just a little map of where this risk is in the world. And this is a heat map where the red means there's a lot of Aedes aegypti, basically, and a lot of risk. And so you can see South America. You will recall from both the Olympic Games and the World Cup in Brazil, considerable concern for the athletes being exposed to mosquito-borne diseases, because it's a very high burden in Brazil and Colombia and various parts of South America, very high level of risk in Africa and India, of course, and Southeast Asia, a little bit of Australia. And you can see it's just, if you look at the map, you can just see it's creeping into the United States. If this map had been drawn 25 years ago, Florida, which is on there, you should know where Florida is because it's the hurricane right there right now. That little red mark, that little red bit sticking out of North America, that's Florida. And they have Aedes aegypti, and they have dengue locally now transmitted. Not in the UK yet, so don't need to panic. No dengue in the UK yet, because the color of this map is inching up every year because of global warming. So Aedes aegypti loves global warming because the conditions for it to thrive are slowly becoming more and more a greater part of the world. So not yet in Europe, but it's probably 10, 15, 20 years ago, there'll be Aedes aegypti in Europe unless we control global warming. Okay. So how do we intervene? And so dengue's been around a long time. There's a lot of interventions. Referring back to the World Cup and the Olympic Games, you probably saw on the TV people with white suits on going around sort of with sprays. They've tried that for COVID too, but they do it a lot for dengue and for malaria, not for sort of cleaning purposes, but to actually, those sprays contain larvicides, which try and kill mosquito larvae in the environment. And they try and remove habitats. It's extremely difficult with Aedes aegypti, actually, because the way I always describe this is, you know, those little bottle caps, this little fellow here on the top of the water, right? So you drink your soft drink or your water, and that cap gets tossed into the street. That cap can breed millions of dengue, of Aedes aegypti. The water, it rains, the water collects in the little cap, absolutely perfect for Aedes aegypti. So trying to remove the habitat means removing all of those kind of objects. And it's really, really difficult to do. So larvicides are moderately effective, but have not really solved the issue. Vector barriers, I've talked about bed nets not being so useful. There's also personal barriers like DEET that you can paint on your body if you like. There's vaccines developed, and there is a vaccine for dengue called Dengvaxia, and it was roughly, and did large clinical trials, about 60% effective, which is not fantastic, but it's not bad for a disease this prevalent. Unfortunately, in another story about big randomized clinical trials, there were some cross-immune reactions for people who got infected after being vaccinated. In particular, children were dying having been vaccinated once they got affected because they were part of the 40%. It's not 100% effective. And so the vaccine was suspended in the Philippines, and I think suspended in several places now. There's a lot of work trying to improve the vaccines. There's, of course, a whole other story about GM development of mosquitoes and a whole new avenue of doing CRISPR research on mosquitoes to try and modify their performance and their ability to transmit viral diseases that I'm not going to talk about. the big leaders in this is a British company, Oxitec, that are experimenting pretty much all over the world. One of the early Oxitec things was to change the genetic structure of the mosquito so that it died before it was fertile. And there's been big trials, and I'm not going to talk about that, but I am going to talk about where I came into the story, which is another kind of intervention called Wolbachia. Wolbachia is a bacteria, and I'll tell you just briefly about Wolbachia. It's a naturally occurring bacteria in the wild. It only occurs in insects. So Wolbachia is a symbiotic bacteria. If you don't know what that means, you should, because you have a whole host of symbiotic bacteria inside you right now, hard at work. In fact, as I look out into the room at each person, I can see Vern, but actually Vern has got more bacterial DNA in his body right now than he has Vern DNA. He's more bacteria than he is human. And we're all like that, so nothing special. We're all like that, and in fact, we know from animal experiments, if you take that bacteria away at birth, the mice die. So if the mice don't get those kind of bacteria, they can't survive, and humans would be the same, we just don't do those experiments, thankfully. So we depend on these, and that's why they're called symbiotic. Largely, the bacteria in humans is in the gut, and it helps you digest food, it helps fight off bad bacteria, it helps you prevent other kinds of bacterial infection, and they're hard at work right now from that cake you ate. They're working hard to help you, and that's great. We feed them constantly by eating, and they protect us, and that's great. So bacteria get a bad name, right? All the antibacterial soap. Well, very few bacteria are bad. Most of them are good for you, and we need them. And Wolbachia is like that for insects. It's a symbiotic bacteria, does not harm the insect, but it does do various things that helps the insect, and the insect feeds the bacteria. So it's a win-win situation. This bacteria is transmitted through birth, so it's not through touch or some other form. It comes, if a mother is infected with Wolbachia, the offspring will be infected through the larvae. It doesn't infect your cat. I've always think that's important. People care about, can my pet get this? Like, could pets get COVID-19 was a big thing. Well, yeah, the answer is yes, but Wolbachia you can't, and you can't get it. So it's an insect bacteria. Most insects have it, but the big science behind this story that happened 10, 15 years ago was Aedes aegypti did not have Wolbachia. Some other mosquitoes did, just naturally, for whatever reasons, but Aedes aegypti did not. And then work in some labs, particularly Scott O'Neill's lab in Australia, they spent 10 years trying to transinfect Aedes aegypti with it, and finally succeeded. And once you've succeeded, you suddenly leap and grab that mosquito and breed it like crazy, because it's spread through birth. And now you have a colony of all those mosquitoes having Wolbachia, and then you breed them and you breed them. And I've been there. It's a very fascinating thing watching breeding. They encouraged me to go in feeding session. Like, I thought it was going to be like feeding penguins or feeding otters. It would be really cute. What they meant was I had to put my arm up against the cage, and then the mosquitoes just, and that would be the feeding. That's how you feed mosquitoes in the lab. Not my favorite day. It's a little bit interesting, the science, and this is what I love about statistics, is you get to interact with the science of the problem and understand things that I have really no training in at all. But there's what's known as cytoplasmic incompatibility with Wolbachia. That reflects the fact that you've got a little schematic here of a female mosquito on the left and a male on the right. If a male mosquito who's infected with Wolbachia mates with a female uninfected, no eggs can be fertilized. So the male is essentially infertile, which in and of itself suggests a solution to repressing mosquito populations might be to infect a lot of males and drop them in. So it'd be just like dropping a lot of infertile males. In fact, that is used in California's Central Valley for the protect agriculture, the agriculture interest. There's a slight problem in that it's expensive to find male mosquitoes, right? So you don't just put your hand in the cage and lift the mosquito up and say, yeah, male, and put it, it's actually more complicated. So it's actually a bit of a lab technology expense to try and separate male and female mosquitoes. But that is a possibility, and it is used as an attempt to suppress mosquito growth. But the other two conditions, automatically, whether the male is infected or not, if the female is infected with the Wolbachia, all the eggs are automatically affected. So this is a great selective pressure to allow Wolbachia to spread quickly in the whole population because of that, that selection. So if the males happen, if you have a whole bunch of females, if the males are infected, doesn't matter because they're not going to lay any eggs. As soon as some females get infected, every egg gets infected. And this is a little schematic, just to give you a scale. So if you start dropping these Wolbachia infected mosquitoes in the wild, within about 2 to 3 months, the entire population becomes infected with Wolbachia because of that selective pressure. So this is the idea of breeding these, taking them out. You usually take out larvae, which you know are infected with Wolbachia, and you put them around a community, they hatch, and they start mating in the wild, and pretty soon you go from wild mosquitoes in the red to green mosquitoes, which are Wolbachia infected. And this is actual data now from Northern Australia back in the early part of the last decade, which showed that after about 3 months, this is in Cairns, Australia, you were getting 90 to 100% of infection rates of Wolbachia after the initial deployment. So the initial deployment is just a little green bar at the beginning and nothing after that. So a big attraction to this strategy is you don't have to keep going back and redeploying. Once it's done, it's done. And in fact, this stops this little schematic in 2015, but if you ran this to 2022, it's kept going. So there hasn't really been any depletion of that. In the lab, we could do various experiments to look at what happens when you infect an Aedes aegypti with Wolbachia. So initially it was thought, the idea was it would have some sort of suppression of the lifespan of the mosquito and it might be used as a suppression. That doesn't really work. So everyone in Australia was disappointed. It's a bust. It's a great lesson of science because somebody just said, let's just infect some of these Aedes aegypti with viruses and see what happens. And they did this and they said, oh my gosh, the bacteria stops the replication of the virus in the mosquito. That's another great story that I'm not going to spend, but it's to do with the enzymes released by the bacteria in the gut. The mosquito it ingests dengue from a meal, blood meal. The virus starts to replicate, but it can't transmit from the gut back up through to the mosquito's saliva. So it blocks that transmission. It's a lovely story, but another story, not so much of a statistical story. And so we knew this in the lab. It was reducing infectivity by 70, 80% of all 4 dengue viruses, so not selective. Wow, was the reaction. So that's interesting, right? Somebody, again, you wonder who these people are, thought, gee, if that does that for dengue, does it do it for anything else? Well, it does it for yellow fever. Yellow fever can't transmit, is blocked by Wolbachia. Yellow fever, there's a very good vaccine. If you've ever been to Africa, you've probably had yellow fever vaccine, but it's still endemic in many countries and still a major problem because of access to vaccines. So that's interesting. It not only stops dengue, it stops yellow fever. Hey, it stops West Nile virus, for which there isn't a vaccine. It stops chikungunya, if you know what chikungunya is, kind of unpleasant infection. Also, all of these are spread by Aedes aegypti. It stops Zika, which you all know about because of the Zika outbreak several years ago. So it blocks Zika transmission. Can you imagine if you're a pharmaceutical company and you come up with a drug that stops all of these diseases at once with one dose? So this is amazing. It's a magic bullet in some sense, right? Because it blocks all of these viruses and a whole bunch of others. Not malaria, not all of the malaria, but that's another story. So wow, fantastic. We were in the lab, they've transinfected it. It has this magical properties. Does it work? Right, so it's like developing a vaccine. Does it work? So how do we know if it works in the field? Call a statistician, right? How are you going to design a study? First, you have to get funders and so on, but how are we going to do this? So the statisticians and the epidemiologists started to get together, and it's obvious if you know anything about dengue that most infections in a given year occur in children and youth, because once you're an adult, and certainly once you're my age, in any endemic country, you're going to be immune to all four if you've survived, because you've been infected by all four. So naive infections occur in young children. They get immunity built up in their youth. So you're going to have to study children and young children and youth largely. And it's hard to do that. Adults are bad enough, as many people in the audience know, to do cohort studies. Children are much worse because they're more mobile. They change, you try and recruit them through schools, they change schools, it's a mess. And to monitor their susceptibility or what's happening with regard to infection, you're going to have to do regular blood draws. Well, you're not going to do a blood draw every day, so you're going to do a blood draw every 3 or 6 months, but you're going to have to do this repeatedly. Most of those blood draws are not going to show any infection, because although dengue is a serious problem in a 1 to 2 year period, there's still a small number of kids that get infected all the time. So it's going to be expensive, hard to persuade parents to let you draw blood constantly when there's no infection, and difficult to do. And if you're doing it every 6 months, which might be a reasonable strategy, you don't know, once you see, you detect antibodies to a particular dengue, you don't know actually in that 6 month period when the infection occurred. So you don't quite know what their exposure status was around the time of infection. If you're trying to do an experiment where you're going to try and expose some kids to an intervention and some not to, it's going to be tricky to know, well, what was their intervention exposure right at the time of infection? Okay, so that's the end of the second story. Now they're going to collide, because these stories were going on separately. I didn't know anything about influenza vaccines, but I was knee deep in this dengue, trying to brainstorm how to design a study. And I didn't know about what's called the test negative design. So we came up with a design which is effectively a test negative design, which is the same answer as the influenza. So these two stories have the same general answer, though they differ in important ways. So what's the test negative design? Well, it was developed largely for influenza, but it's now been widely used for things like rotavirus and other kinds of viral, to measure vaccine efficacy. So the idea is I'm going to describe it very simplistically, you recruit individuals in the healthcare setting who have symptoms of the disease that you're interested in, but the symptoms aren't necessarily that specific. The same symptoms may occur for other viral infections or other kinds of disease. So if you're interested in the influenza vaccine, you would be recruiting people who show up in a healthcare setting with upper respiratory healthcare symptoms, and you recruit them. You might measure key covariates at that point. In particular, since you get the individual right as they're sick, you get to ask them, what were you doing last week or the last 2 weeks? Where were you? Who were you seeing? You can, whatever's relevant to the disease you're interested in, you can ask them that. As against the cohort setting, I said, well, you don't know what time period to ask them about, and you get it maybe 4 months after it's happened. And who knows what your kid was doing? If I asked you, what was your kid 4 months ago, you probably can't remember. But now you've got them right there, they're sick. You ask the parents or the individual, where were you, what were you doing, and so on. And then you subsequently definitively test for the presence of a specific viral condition, like influenza. Some of those people who are recruited will test positive and some will test negative. And that's the idea behind the test negative design. You're going to compare the test positives with the test negatives with regard to their exposure. So for the seasonal influenza, you have these clinics, people show up, they've got flu-like symptoms, they get tested, and you ask them at the recruitment, did you receive this year's seasonal flu vaccine or not? When did you get it? And then subsequently they get tested and you compare the test positives with the test negatives. And that's exactly what we sort of eventually brainstormed to do with dengue. We were going to recruit people who had dengue-like symptoms. We would ask them where they had been essentially in the last couple of weeks, and then test for the presence of dengue. And there's very highly sensitive and specific tests for something like dengue, as there are for influenza. So what are the advantages of this design? One is, and I'll come touch on this in the next slide very briefly, but you're able to remove or reduce at least confounding due to healthcare seeking behavior. So people who have high propensity to seek healthcare tend to get vaccinated a lot more than people who don't have a high propensity to seek healthcare. Well, it makes sense, right? And they also show up at clinics when they get sick more. So there's a real potential for confounding that would show that vaccination is bad, because the people showing up to be tested tend to be ones who've been vaccinated. And so this gets rid of that, because everyone in the study is showing up with similar symptoms. And there's a little bit here I'm not going to touch on that is, well, how do you pick what the definition of the conditions are? How broad a set of healthcare conditions can you do? And that's relevant to this question. You get to measure what they were doing at the time of disease rather than in the past. And it's a very efficient way of statistically. So now, of course, there's some key assumptions, and I'm going to just breeze through these very quickly for the sake of time. The key one is that the intervention you're interested in does not influence other test negative conditions. So you've got an assumption in here that the flu vaccine does not make any difference for any other upper respiratory infection. Actually, not an unreasonable assumption. The flu vaccine is very specific. It does not protect you against other upper respiratory tract infections. But you can see in the dengue study, we had to test for Zika and chikungunya as well, because our intervention did protect against those. So those people they could not be test negatives. So that's a key assumption. And once we got rid of those, the assumption is that Wolbachia has no influence on influenza or other febrile illnesses. It's fine that the intervention or exposure might influence enrollment. That's what you would expect with vaccination, or in our case with intervention. People might show up to the healthcare setting more frequently because this is unblinded. They know they've been exposed. That doesn't matter as long as that association isn't differential depending on whether they have the condition you're interested in or not. If it does, you've got a real problem. But remember, these people show up before they know what they have. So in principle, how can it influence their decisions? Because they don't know if they've got influenza or not, or dengue or not. So that's important assumption, but the design really accommodates that. And that's important for, as I said, for internal validity. There's an external validity question here, which is that the efficacy of the intervention or exposure, you're only going to be able to measure it in a healthcare seeking population. You never see people who don't seek healthcare for these conditions. And so that may be a sizable amount of the population. You're going to have to assume. So when you see that in the BBC, about 60% of this year, that's actually extrapolating to assume that applies to everybody, because they only measure that on people who showed up at a clinic with symptoms. And there is, of course, you want a good test to be able to differentiate test positive and test negative. There's lots of statistics done to try and accommodate misclassification, but I'm not going to get into that. Okay, And so test negative designs were around for influenza. I'll come back to COVID very briefly, because now the rubber hits the road for the statistician. I've hopefully given you a general idea what a test negative design is and some of the general big assumptions, but the devil's in the details, as it always is, about how do people get recruited? How do you measure things and so on? So there's a lot of subtle details. There's work for you guys, is what I'm trying to say, to think about this. New design appears, looks nice, looks simple, but it's important that statisticians really look at things carefully. So as I said, it significantly reduces the bias due to healthcare seeking behavior, and I put a DAG up there. So now I'm sort of looking at the causal inference people. I know there are many in the room who know far more than I do. So there's a nice DAG, and there's the sort of classic confounding picture there, healthcare seeking behavior causing, increasing the chance of being vaccinated, V, and also increasing the risk of infection. You got classic confounding, but if you condition on holding healthcare seeking behavior fixed, you remove that. That's the rough, that was the motivation for the test negative design. This is very simplistic, and Brahmar's in the audience. I know she's thought about this a little bit. This is overly simplistic, in part because healthcare seeking behavior is not a binary measure. It's not 0, 1. You've got it's sort of on a scale. There's a propensity to seek healthcare. And so it's a little bit more complicated than this. That's why I'm appealing to the causal inference people in the audience. So it can get quickly out of control. And even these DAGs that are on here don't entirely capture, in my view, the role of the test negative condition and how you define that. If the test negative condition was having a broken leg, it doesn't work because everyone seeks healthcare with a broken leg, I assume. And so that doesn't work, because when you look at vaccination status amongst people who break legs, you're basically getting the prevalence of vaccination in the population. But when you look at vaccination status for people who are coming in for COVID, you're going to get people who are, you're going to get a higher estimate of vaccination coverage because of that connection of vaccination with the propensity to seek healthcare. So it's all I'm saying is the causal inference is complicated. You guys need to solve it for us all. And there's some very interesting work that I've been talking to Eric Chetkin, who's at the University of Pennsylvania, and some of Bramar's colleagues at Michigan are involved in this work too. And it's to do with what Eric has called double negative control status. So I'm not going to go into any detail. I'm just in it to tantalize you. It's a little appetizer for those of you trying to stir up interest. So a double negative control study, it has two controls going on. There's a negative control exposure with the assumption that that exposure has no effect on COVID, or that exposure has no effect on influenza or dengue, whatever your disease is of interest. That's a negative control exposure. I've already talked about a negative control outcome, which is the core basis for a test negative design. That's the outcome that has no relationship to your intervention or exposure. And some of Eric's work and this group at Michigan is now showing that if you use both of these simultaneously, you can actually remove some of the potential hidden bias that's illustrated by these very complicated DAGs on the right-hand side of the screen. So I just, again, putting out that there's lots of interesting work to be done. It's very connected to what Eric calls proximal inference and the use of controls in epidemiology, which has been underutilized to a large extent. So as I said, I don't have time to go into detail, but I wanted to spend, I have about 10 more minutes, is that? But I haven't been following. Who's in charge here?
20 minutes.
Okay, great. I want to just do a little bit about analysis now. So this has all been about design, but hopefully you see it's really important that people like you get engaged in these design questions because they're really fundamentally statistical. So what about analysis? So here's the original test negative design analysis strategy. So this whole table is supposed to describe people who will seek healthcare if they were to get symptoms compatible with the disease of interest and whatever you define test negatives to be. So the NI and NC are The rows here are people who have the intervention or not. That might be vaccination. It's not going to be that for my dengue trial, but for the moment, just can think of that exposed or unexposed. So what you see is what's in the red box, because those are the people who show up with symptoms and they either test positive or test negative. So that's what you see, and you measure A, B, G, and H, but you don't know NI and NC. And in fact, it's quite difficult to estimate NI and NC because it's sort of counterfactual. It's if they were to get a disease, would they show up? How do you measure that, right, in the population, the size of that population? Because you really would need to know those to get the relative risk. So the relative risk is just the infection rate, the test positive rate in the people exposed compared to the test positive rate in the people unexposed. But you don't know NI and NC, and it's hard to estimate. But here's the key where you use the test negatives with the assumption that the intervention has no relationship to the test negative status to estimate NI and NC, or the ratio specifically. So you assume, by assumption, that the rate of the test negative condition is unaffected by the intervention. That was the key assumption I had on the previous slide. So the rate should be the same. That means B over NI is the same as H over NC. That's that assumption encapsulated in the data. And now you can see we can estimate the ratio of NI over NC by B over H, which we do see. And lo and behold, you get the odds ratio. So this was probably obvious to all of you. It took me a little while to understand all this stuff. So you can just take the odds ratio, and actually that odds ratio estimates the relative risk. Because this study, for those of you who are trained in classic epidemiology, this study really approximates a case cohort study than a true case control study. Because the rate of intervention or exposure in the general population is what's captured by the test negative column. They're not true controls. Those people may ultimately become positives. So that's the relationship between case cohort and case control study. The bottom line is you can use the odds ratio to estimate the relative risk and do all your old odds ratio. As I said, this is complicated. It's not as easy as just computing that odds ratio in COVID-19 studies because there are lots of things going on that we sort of heard of during the day. For the outcome misclassification here is not about the test, the PCR test for COVID, but it may be that individuals continue to shed, as we know. We heard a talk earlier this week at the unit from it's Josh. Where are you? Josh's talk about how long you may be shedding and detect as positive once you've been infected. That may go on for quite a long time. And so you may show up with another viral infection to the clinic while you're still shedding virus from a previous COVID infection and be classified as a test positive when you're really a test negative. And so it's a little bit of a problem, and there may be a misclassification in the opposite direction of asymptomatic cases may be misclassified as controls. So you need to understand the subject matter, the actual viral condition to understand this. And the sensitivity and the specificity of the testing assay becomes an issue. And in fact, in this case, being too good may be not great. You wouldn't want an assay that could detect ever the presence of COVID-19 from 4 years ago because of the problem I just indicated. So it's interesting. Other things, and some of this is push people doing tests, and test negative designs, by the way, are being used all over the world right now to estimate COVID vaccine efficacy. And sometimes it pushes people to use harder clinical endpoints like hospitalization. There are other problems with COVID-19 I'll touch on briefly just at the very end. And now we've got the issue of background immunity and how we can assess that and how that affects things. And again, Bermar's group has written some commentary on this a little bit, and there's some papers trying to grapple with applying this into COVID. And here's just an example of one I was involved in California. So this is the kind of picture you get at the end. You've all seen these pictures on Twitter, if nowhere else, right, of the vaccine efficacy. This was fairly early, and these were Moderna and Pfizer because that was all that was available in California. You can see this is pre-Omicron, the efficacy and measuring efficacy by how many doses and other conditions. Okay, back now to the second part of the story is, what's the test negative design got to do with Wolbachia? So what we did here was think of doing a test negative design in exactly that way, but how was exposure going to be allocated? Because Wolbachia mosquitoes are not issued individually like vaccines. You can't go and buy one and carry it around with you. So it's clear, for those of you ever been involved in vector control studies, you're going to have to do some sort of cluster study. So we did cluster randomization. And this is just a picture. This is a picture of Yogyakarta in Indonesia, which is in Java. It's a big city. I always say it's the size of Oakland, which is fine for anyone in California, but you guys don't know what Oakland looks like, but it's a big city. And not like Jakarta, but it's big. It's very Indonesian style city, urban living. And here we divided the city into 24 regions and randomly allocated the intervention to 12. And they're the ones colored in green. And that's a huge story in itself, a lot of community engagement, because what were we going to do? We're going to be coming around your house and putting a bucket of larvae right outside that are going to hatch mosquitoes, and it doesn't sound like a great thing. So there's a lot of public engagement and involvement involved in this study. It was constrained randomization, another story I'm not going to talk about, because there's not that many clusters. So how does this compare to the influenza vaccine or the COVID vaccine test negative designs? Well, two major statistical differences. One is the intervention is clustered. So we've got aspects of cluster randomized trials. And two, the exposure is randomized though. We had, the statisticians were able to get in there and impose some level of randomization. So less chance of confounding. So it's sort of like a randomized cluster randomized trial, but with case control surveillance to measure outcomes. There have to be other examples of this, but I've never seen another one. I just figure there has to be somewhere where somebody had a randomized exposure, but for efficiency reasons, measured the outcome in a case control fashion. So this is essentially, and we didn't know at this point when this was designed, we didn't know the words test negative. It just so happened, it's like all great ideas, it's been thought of before. Here's the data that you see. The data is now a little different from what I just showed you from a general test negative design, because you get the same rough idea here. This is what you see in the box. This is for each cluster. J is indicating cluster here. But in each cluster, one of those rows is 0, because the clusters are all receiving the intervention or all not. So you can't get a cluster-specific odds ratio. It's just not possible. So, so you're going to have to use those rows. You'll either see A and B, or you'll see G and H, depending on which cluster you're in. And from that, you have to do statistical inference about the effect of the intervention. And you've got to account for the clustering. And you really want to exploit the randomization. And you can do that with permutation tests. So if any of you know about randomization inference, this is a really nice example of how you can use it, because there's only 24 sampling units. And you can, if you know about finite sampling, you can develop approximations to the permutation test, which are very useful for simulations, because running simulations of permutation tests are very difficult because they take so much computation. And here's the results of the trial. And I hate to do that slide transition, because this was 5 years of blood, sweat, and tears to get to this point. But this is the fun part, right? With all that work as statisticians, you finally get the data. And here was the data. It was extraordinarily effective, almost 80% effective in reducing infections, and equally effective for all the serotypes, and even more effective in protecting against hospitalized dengue. I think I sort of swept by there. So you know you can look at the aggregate odds ratio is still there, even though each cluster contributes one row, either the top row or the bottom row. But you can aggregate, you get an odds ratio. It's based on cluster data, so you have to worry about that in the inference. That's why permutations are useful, because they maintain the cluster membership. That's what these numbers were based on, was on the odds ratio, the aggregate odds ratio, accounting for that clustering using permutation. inference and using those approximations to get confidence intervals. You don't get a visualization of what actually happened. It's kind of dissatisfying, right? You just get those 4 numbers, the aggregate odds ratio, and then you get a black box of things that you and I love to do, but no one else knows what's going on, where you're permuting and doing all this stuff. And out comes a confidence interval and an estimate. And the investigator said, yeah, but how did it work? Which clusters were it really working? Right, so you need a visualization, and you can't get it from the odds ratio. So we came up with this idea, which is the proportion. You see, in each cluster, you have some test positives and you have some test negatives. And you look at, well, what proportion of all of the recruits were test positives? And you plot that for each cluster, and there you can see that you can visualize. And there you can see the blue is the intervention proportion. So most of them, very few of them were dengue, compared to the control clusters where a greater fraction of the recruits were testing positive for dengue. And you can see only actually one cluster there crossed that gap between the intervention clusters and the untreated clusters. Of course, that's great for visualization. Can you also use that plot to estimate efficacy as an alternate to the odds ratio? Well, yes, you can. Because you're all statisticians, you can look at, well, what's the average of that proportion? If you think of sampling these clusters in the intervention, and what's the average for the controls, you can sort of see there a little confidence band and box plot there of the average and a confidence interval. So can you compare those? Yes, you can. Kind of like a t-test. You're just going to do essentially a t-test on those averages. That t-test with a little bit of high school algebra relates back to the relative risk through this quadratic. I'm not going to go through the details. It's not hard. And then you can, R is just the ratio of the total number of test negative controls to test positives. You can solve, once you estimate T, you know R, and you can estimate lambda. And that actually gave almost identical answer in this case. And there's a lot of simulation, looking at which of these works well. Very high efficacy. I think I'll just go over, I won't go over the slide, but there's a new one. This estimator that we originally came up with, I talked about it doesn't matter if there's differential healthcare seeking behavior comparing the intervention controls. And in this trial was unblinded. So there was a huge citywide event, like the World Cup, where we drew the balls out of the boxes to engage the community. And so everyone in the community in principle knew, do I live in an intervention cluster or not? And I remember when I visited, my first question when I landed was, is the hotel in an intervention cluster or not? It wasn't, but anyway. So everyone knew in principle, and it's fine, in general it's fine if that's differential, but later we observed you can introduce some bias if that differential behavior is further differential across the clusters. You argue whether that's plausible or not, but it does introduce bias, and there's a slight variant to what I just did, where instead of taking the average of those proportions, you take the average of the log odds. That is unbiased. And in this case, I won't go through the details, but it gives a very similar estimate. So probably not much bias in this particular setting. And then there's other things that you guys all love and justify our jobs, where you can, well, what about adjusting for cluster level covariates, et cetera, et cetera. And yes, you can do that, and you can get a more precise estimate if you have good informative cluster covariates. So let me just add one more little part to the story, is to, now I've talked to the causal inference people, a lot of work to do. I've talked to sort of inference, randomization, cluster randomized trial people, a lot of interesting things to do. Now let me turn to the spatial people in the audience, people who are interested in the spatial distribution of disease. Here is the spatial distribution of all the test positives in the trial, and they're color-coded by the serotype. By the way, if you have dengue 1, you can't, and a mosquito bites you and then it bites your partner gets dengue 1, they don't get dengue 2. They're serotypes, they're 4 different viruses. They're in the same family, but so the color is kind of important. I've ignored the color, which serotype it was, except I did give you the individual odds ratios. But here's the spatial distribution. You take a look at that as a statistician, you can pretty quickly see, you don't know the population density here, so, and I haven't showed you the controls, which give you a sense of the population density, but just looking, eyeballing that, it does look good, right? The gray, which are the intervention clusters, don't seem to have as many dots in there. That's a little distorted because they say you don't know, maybe the gray ones are where hardly any people live. So the controls are important, so don't get me wrong. Then we looked at, well, but how many of those dengue cases are close to someone else who has the same serotype and was infected within the last 30 days? You can change that definition as you wish. Did I run out of battery? This is the most elaborate scheme to get the speaker to shut up and sit down. We're literally 5 minutes away. You want me to wait?
Just 2 minutes.
Yeah, you've got to turn the volume up. Oh, that's picking me up. Okay. Okay, so on the right-hand side is the distribution, but now in serotypic, what do we call homotypic pairs. So these are pairs of individuals infected with the same serotype, 1, 2, 3, 4, within 30 days of each other. So it's capturing what we know about dengue is, of course, it's spread from individual to individual in close regions. The most likely person you'll get dengue from is your sleeping partner, because the mosquito will bite them. And if they're infected and they're more likely to bite you, then that puts you at risk. And now if you look at that picture as a statistician, you can start to see, okay, I know there's less dots in the gray areas, I already said that and tried to have to be careful about interpreting that, but there's far less homotypic pairs in the gray areas, even more so than the dots. So that suggests looking at the question of, does the intervention impact not only reduce incidence, but does it interfere with the clustering of dengue? So you will get sporadic cases of dengue in an intervention area because people move around and mosquitoes move around, and people get dengue from the next city over on the weekend and then come into the city and then get sick. So you will get them, but the question is, will they cause a little mini outbreak, a cluster, if you're protected with Wolbachia? And the answer is yes. Wolbachia not only reduces the incidence, but by using spatial clustering techniques developed by statisticians, we're able to determine, yes, there's a difference between the clustering in the intervention clusters than there are in the what we see in the control clusters, adjusting for the incidence level being already different. So it's statistically here, normally, I mean, people like Peter Diggle see these pictures all the time, right? This is Peter's bread and butter. Spatial distribution of childhood leukemia. If you read any of Peter's papers, you'll see a picture like this. There's rarely a picture of controls, but the beauty of the test negative design is the controls come along for free. And you have clustering of the controls. Now you might think the controls also might be clustering because there are other upper respiratory viral infections also have some sort of level of clustering. But you might have a different level of clustering. So here's an appeal to the spatial people, and there's work to be done here to think about these things, about how you would use the controls to assess the clustering. There's a slightly different question that is also related to work of Peter Diggle, which is, what's the effect of my risk if I live within 300 meters of someone who's had dengue in the last week? And we can measure that a little bit with the data, and you can build up what's equivalent to a Cox partial likelihood, but it's got a spatial aspect to the risk set. To be in the risk set, you have to be, you have to look at who else is within 300 meters of you in the data set. But you can build a partial likelihood and measure the question, what's the impact of having someone close to me who's had dengue recently? And does Wolbachia change that risk? No, it doesn't. Overall, it changes the risk, but it doesn't interfere with that level. This is a slightly different question than the spatial clustering, but more work to be done. So the story is not ended. And I've tried to argue with, there's a big step wedge community, particularly in the UK, funnily enough, but also in the US a little bit, people who are really promoting step wedge designs. There's a version of all this for step wedge designs, of course, and actually in Colombia, they wouldn't let us do a parallel arm study. That's that experiment randomization version. But I asked them, okay, we understand you don't want to let us randomize our allocation of Wolbachia to communities. This was around Medellin. But I very cheekily but quietly kept a straight face. Do you mind how we roll it out? Does it matter to you whether we roll it out here, then there, then there? And I said, no. So I randomized how we rolled it out without telling them. So we had a step wedge version, basically. And the beauty of the test negative design, you have these controls there, which most step wedge designs don't have, right? Step wedge designs haven't really bubbled into their thinking the idea of using either a negative control exposure or a negative control outcome. And they're very useful for getting rid of residual confounding. Okay, final couple of slides. I appreciate your patience. So all of the above, you may have been sitting there and you may have a question ready to go, but wait a minute, this assumes that people show up in the clinic to be tested because they have symptoms for the same reason. But we know that isn't true for COVID. People don't get tested for the same reason. Some people get tested asymptomatically. So at Berkeley, we forced students to get tested asymptomatically twice a week because we didn't want outbreaks in the dorms. That's different. So there's more than one route to be tested. Is that a problem? The answer is yes. That's a real problem statistically. And this came up before COVID because I was working with Ebola design for testing vaccine efficacy of a new Ebola vaccine. You may have heard of an Ebola vaccine that was developed after the West African Ebola outbreak. There's another vaccine that was being tested in the DRC, in the Congo, and they used at LSHTM, and they used a test negative design. So people would show up with Ebola-like symptoms. Some of them are positive, some of them are negative, and you know the story now. But Ebola's not that, Ebola-like conditions and other conditions that have Ebola-like symptoms aren't that common, thankfully, right? And so what happens when you show up and say, I think I might have Ebola. All right, we're going to test you. They don't just stop there. They say, who are your contacts? Who have you been in contact in the last 3-4 days? And they immediately test all of those people and try and isolate anyone who tests positive to get them removed. So you have another set of people who are being tested that are linked to the original person who showed up, who are being tested. And original design at LSHTM, this was basically Peter Pio's group, was going to throw those away because they knew it was bad to include them as if they were the same. But there's a lot of people there at high risk. And you get actually more Ebola cases in those set of contact because we know they've had a high level of exposure to an Ebola case. And the question is, statistically, get to work. How do you combine both of these groups? So in COVID test negative designs, how do you account for the route for testing? Was it symptoms or were you asymptomatically screened? Here it is in a slightly different setting in Ebola. And it's interesting in Ebola because the vaccination status, whether you've had the Ebola vaccine or not, is likely linked for the person who shows up with symptoms to their contacts. They're not unrelated, right? And the contacts may be in their own family and so on. So it's a slightly complicated statistical question. And so there's lots of work in development. Okay, the last slide. So what I've hoped I've conveyed is my passion, and there's lots of interesting and challenging statistical problems. The moment you get an infectious disease and you start unraveling things, lots of room for statisticians, lots of room in design. The ring design for the original Ebola vaccine was an interesting vaccine trial design. And by the way, we need you. There's a new Ebola outbreak right now in Uganda. And it's not, you're not protected with the current vaccine. It's the Sudan version of Ebola, and the version that was the West Africa does not protect you against the Sudan, and it's rapidly spreading. So don't panic. Not going to spread to the UK probably, but it's a real concern that it doesn't grow to a West African size epidemic, and we're right at the cusp of it at the moment. Lots of interesting analysis issues that you don't really learn about if you're exposed to chronic diseases because of the dynamics of infectious disease transmission. Very complex causal mechanisms because of the feedback loops that are inherent in infectious diseases. And you learn about, oh my gosh, there's an outbreak, you change your behavior. That doesn't tend to be an issue in cardiovascular disease in short term, because the time scale of infectious disease is so different than chronic diseases. Lots of missing data. We heard that from all the talks this morning, the missing data, because we don't observe when the infection actually happens and when certain things happen. So lots of, and Sean was reporting delays, lots of filtering of data that goes on. Lots of interesting statistical questions about what the right contact tracing algorithms would be. And I won't go into the whole test and trace fiasco in the UK, but lots of interesting questions to design those. And different contact tracing algorithms actually were used in Southeast Asia than we tried to use in the UK. Lots of interest about, Sophia's here somewhere, and the graduate students are working about group testing. Lots of interesting ideas. Well, testing can be expensive. How can we statistically exploit our knowledge by pooling samples to test and so on? Why don't we teach our students about infectious diseases? There's not a single course at Berkeley when I arrived on the statistics of infectious disease. There were a whole bunch of courses on cardiovascular epidemiology, cancer. We don't really do it that much. And I hope that COVID-19 is changing that it's showing that there's a real room to we need trained young people who are familiar with these concepts and ideas and can step into the breach quickly. I'm not saying we don't need better models. Fantastic to get better models. Lots of people have been doing in that. But my plea in every COVID talk I've ever given is we need much, much better data. And we've crippled ourselves. And I'm a bit US-centric here. Totally crippled ourselves by having no data. We have been flying blind the entire 2.5 years, and that's led to enormous public confusion. And it's partly because we don't have good data systems. And we need to think now about what data systems should we use, given that the next pandemic, I've lived through 5, so number 6 is just around the corner, won't be probably COVID, won't probably be a coronavirus, it'll be something else. So we don't want to design data systems that are COVID-centric, but we need something to think about that will detect and allow us to respond with interventions quickly, whatever the next pandemic is. Thank you very much for your attention.
Okay, we have the microphone. Shall we take, so people online, please stick your hand up if you have questions. Whilst you do that, thank you very much, Nick, that was great. And we'll take 1 or 2 questions from the room while the online people warm up.
I'm throwing it. Hey, thanks very much. That was really interesting, especially like the stuff on dengue as a former Aedes aegypti breeder. So I was wondering, so in terms of the practicality of the study, you're introducing these Wolbachia infected mosquitoes into these areas. Did you have to run the study extremely quickly so you didn't get Wolbachia contamination into your into your air quotes clean areas? And if so, how did that affect the way that you designed it?
Yeah, that's a terrific question, and it always comes up because anyone with any knowledge about cluster randomized is used to villages that are separated by hundreds of miles to avoid contamination, right? And here, we're trying to do a cluster randomized trial in an urban setting where contamination is inevitable, right? So this is also a little bit different and threatening statistically. We couldn't do it quickly. First of all, we didn't start disease surveillance until the Wolbachia prevalence hit a predetermined peak. And by the way, Wolbachia prevalence was being monitored throughout the entire trial with little traps, with pool testing, by the way, to measure Wolbachia prevalence. So we waited until it got in, and then we started the clinic recruitment. So that took a couple months. But that's not really addressing your question. But then we had to wait. Of course, there were a lot of sample size and power calculations. How long do we need to run this trial? It was supposed to be 3 years to do this. Actually, what happened was COVID, of course, happened about year 2.3 or 2.4, and the clinics all closed, and we had to end the trial. Thank God it was so efficacious, it didn't matter from a power point of view. The contamination issue was by then, though, quite real. So people move for a start. So kids move to their grandparents at the weekend or whatever, and they move from a protected area to an unprotected and vice versa. They go out of the city. So part of the recruitment involved a detailed questionnaire about movement in the last few weeks so that we could actually do a per protocol analysis ultimately of a weighted average of how much of the time in the last 2 weeks were you in a protected area and so on. We also had the Wolbachia prevalence in the immediate surrounding your home, because these traps were scattered throughout the city. So we had the ability to do that. And the clusters were designed deliberately to include highways and rivers to try and avoid contamination. So there was a lot of thought that went in there. And mosquitoes don't wander far, thank goodness. So they live, if you did nothing to them, probably about twice the size of this auditorium as far as a mosquito travels in its lifetime. But there's wind. And if mosquito goes up, a gust of wind does it. So eventually contamination, we were able to detect it because we were trapping all the time, and we had to modify the protocol about year 2, recognizing that some of the control, there was no problem with the intervention areas dropping in their Wolbachia prevalence, but 2 or 3 of the control clusters started to show 10% Wolbachia prevalence, 15%. So we modified the protocol to end the trial if a certain number of clusters went above 20 or 25%, because it was just going to dilute the efficacy estimate. So that was a real concern. We just, I think, to answer your question, just squeaked in time. I think if the trial had gone on, and it probably would have ended early just for contamination reasons, but it was a threat.
It sounds like an incredibly complex, well, just practically, it sounds very, very impressive.
I remember when we designed the trial, many vector control people publicly said, this is a disaster, it will never succeed, for a whole bunch of reasons. It's very hard to do vector trials where you have a human health as the outcome. A lot of trials about mosquito abundance suppression or things like that, that's the typical mode in these vector control trials of saying, my shotgun that kills mosquitoes, and they measure how many mosquitoes, but they don't know what level you need to get mosquitoes down to make an impact on human health. So this was one of the very early, if not the only trial I know of this kind. There was one with Oxitec was running, but they had human health outcomes, because that's what we really care about. But it was widely disparaged at the beginning by the vector trial community as not being possible. And the Gates Foundation didn't want to fund this because they thought that it was too risky, that it would be negative, and that which funder wants to be associated with a failed trial? So yeah, it was a challenge. So you can imagine my scream when I saw the results. And there's an interesting story about this for people who serve on DSMVs, because, you know, there was a DSMV or some mnemonic equivalent, people watching the trial. And part of the protocol said they would report hospitalizations and deaths by cluster. So you would imagine do that, right? You just make sure your intervention isn't killing people for some reason. Maybe we were releasing, you know, maniacal Wolbachia mosquitoes. Who knows? Halfway through the trial, the DSMB said there's more hospitalizations in the intervention clusters than there are in the control clusters. And so I got a panic call from Australia saying, oh my God, how can this be? And I said, and actually I was in London, I think at the time, and talking to Emo Kleinschmidt, who's also at the London School. And after about 5 minutes, he said, oh, that's great. Of course there should be less. They didn't know, sorry, I should rephrase that. There were more hospitalizations in one of either the intervention control, but everyone was blinded to which was which, except me. But the DSMB said that, and they said there were real concern. And I'd say after 5 minutes, they said, well, yes, there should be more hospitalizations in one or the other, hopefully in the control clusters. And so you can imagine my scream when I got the data, and you know, I was the only one who could unblind it. And yes, I tried to confidentially feed back to the Data Safety Monitoring Committee, you have no concerns, basically. And they knew what that meant. So I did have an early warning that it was working because of that DSMB. It was kind of interesting. And then, of course, it finally came out. So it's huge. I mean, it's a huge impact. So they're all over the world now. We're trying to get governments to invest the high cost initially of deploying these mosquitoes, because for the very reasons we were talking about, mosquitoes don't travel far. You can't just deploy them at one spot in the city. It doesn't just, you have to deploy them everywhere. And then you can go and leave, but there's a high upfront cost. And then the idea is you're protected against Wolbachia for 10 or 20 years or whatever. We don't know yet.
I mean, I was going to ask the same question, but from your answer, it does seem that, if anything, those estimates are an underestimate of what you would get if you could do the whole, you know, global deployment of this, not global.
Yeah, so that protocol analysis came into that, because there we were able to say, well, but some of the controls had a little protection. Yeah. And we had this mobility measurement, so we did it per protocol. And for the statisticians who are interested in cluster randomized trials and generalized estimating equations or generalized linear mixed models that you might do to use a continuous measure of exposure is quite interesting. It's quite complicated, but and I've just finished that work, and the bottom line is it looks like per protocol it's about 90 to 95% effective. So, but it's a tricky calculation to do, but yes, it will. And we knew up front that this was going to ameliorate dilute the efficacy a little bit because of the contamination issue.
So thank you, Nick. Fantastic talk. I especially appreciate your invitation and encouragement for statisticians to get involved in these different problems. I had one question that the testing practice has changed dramatically. People are testing at home, even not testing. So people who are getting RT-PCR tests done, at least in the United States, have a very special kind of mechanism now than 6 months ago. People who are sick or for COVID or with COVID. So how do you see designs evolving when we are comparing, say, primary series vaccination with booster under this type of testing?
Yeah, that's a real threat to test negative designs for COVID, is the testing landscape has changed quite dramatically. Now, I've tested myself multiple times, but never it's reported. And even if it had been positive, I probably would never have reported it unless I got very sick. So it is a threat. I haven't thought deeply about that. I think it's connected a little bit about having to broaden the perspective to look at perhaps different forms of testing data and different, as I said, different routes to why you're being tested. and that somehow you're going to have to combine those. And I was truthful. I don't know the answer to that. It's work in progress, but I think it's really important now. It's like the test positive rate that was paid so much attention to early on was very informative. Now it's worthless in my view. They're still reporting it. I designed a dashboard at Berkeley to monitor and all these asymptomatic tests, and they were reporting the test positive rate, and I was watching that quite a bit. Now it's about 12 or 15%, which would have frightened me to death 2 years ago. But now I think, oh, it's the only people being tested in the health center are people who are really sick. And so, of course, the test positive rate. And there's a whole bunch of people out there that are testing and are negative or that I don't know about. And so, yeah, it's a real challenge for statisticians, I think, to think about that. So this design, it's like case control designs. It's so seductive and simple, but there's a whole raft of real statistical issues to be addressed. There's really currently trying to get involved in monkeypox. And of course, we have a vaccine for monkeypox. And then there the question is a little bit is the vaccine differentially affected? It's mostly happening through sexual contact right now. But what kind of sexual contact? And so how do we get at that the vaccine may have differential efficacy depending on the nature of the exposure? No one's thought about that, of course, for COVID. It's probably not relevant for COVID so much. What kind of exposure? Would a vaccine protect you against some exposures but not against others? Probably not quite so relevant. But for monkeypox, it might well be relevant because that involves more physical contact than just, it's not through the air transmission very much. So lots of interesting questions. Come and work on them. I'm about to quit, so we need replacements quick.
Is there anyone online? No. Let's do Sheila.
Well, that was a terrific lecture, Nick. Thank you very much. Traditionally in the United States, something like the Veterans Administration or Kaiser Permanente has helped in terms of record linkage and so on. So is there thinking in the States about, as you say, the next pandemic and how you can get data acquisition either in a single state or I mean, that may be the best solution that could be hoped for.
Yeah, that's a very political question, Sheila. Kaiser, I worked with Kaiser Permanente in early 2020 because they were very concerned about their ability to accommodate their patients. Kaiser Permanente, by the way, for those of you who don't know, is a large private health insurance company that they provide health insurance in hospitals. So when you sign up for Kaiser, you go to those GPs and those hospitals for care. And it's separate. I can't go to a Kaiser. I could, but I'd have to pay because my health insurance is not related to Kaiser. So it's private. It's a big private company, and they were very concerned and very responsive. They probably cover 1/3 to 1/2 of California. So they're a big player. And they gave us access. Joe Lunard and I got access to their data in March, April 2020 to try and project the healthcare demands on the hospital system a month or two out so they could prepare. And it was terrific, and we got some very early picture of the epidemiology of, people moving to infection through the hospitals, what happens to them and so on. It was a hell of a job publishing that because Kaiser it went all the way to the governor of the state of California before we could publish it. We had to get approval from the governor's office because the governor's office was trying to manage the thing. So it was very complicated and political. I don't mean in a bad way, but it was just complicated. But the state of California complained Kaiser was not willing to share their data. I know this is being recorded, so I have to be a little careful, but my understanding was there was restrictions, at least, at sharing their data outside of the Kaiser system. We had to sign all sorts of confidentiality stuff and get approvals and so on. So even within California right now, there's fundamental obstacles of that data sharing across even hospitals. So there's multiple hospital groups in California, all have their own data systems, and largely not a good incentive for them to share, even in the face of a statewide crisis. So what you ask, what's the answer to that? Gee, if I do the answer to that, I don't know. I mean, obviously the obvious answer as a British person is the national healthcare system. And Obamacare originally had a very deep, because there were a lot of statisticians and health economists involved in developing Obamacare, and I don't want to bore the rest of you, don't care about American politics, but there was originally in the Obamacare what was called a public option, which was basically any member of the public could go online and buy healthcare insurance. And a big component of Obamacare was completely ignored in reporting about it, was healthcare linkage. And the idea that we'd get a huge part of the population signing up for, and small businesses could sign up for Obamacare and so on, and all of these records would be linked. The public option got jettisoned in the debate in the Senate and the House before Obamacare first got, but a large fraction of the public option. And so the debate currently about Obamacare in the United States is progressives trying to sort of re-put in the public option more, but it's a very complicated story, and I'm far from an expert, but I don't see a solution in the United States. We spend an absolute fortune on healthcare, and it's just down the drain because there's nothing, no linkage, nothing. I mean, Bramar may have, you work in electronic health records stuff, so I don't know if you have a better answer for Sheila, but I don't.
I can go on for the next hour.
Yeah, yeah. It's a complicated story. So thank God for the National Health Service.
Yes.
For all its problems, and we've heard about some of the data from it. We don't have that in the United States. It's a real crippled.
No, you're in here.
Not perfect, but it helps. You've got a long way. Yeah.
There's always room for improvement.
Yeah.
Sure. What more questions? Yes.
So if this was set in the relatively simple world of drugs trials and a cardiovascular disease, you'd now be embarking on a phase 4 long-term safety study. So, but my mind boggles that. Can you do that? How does it work?
Are you talking about the mosquito trial?
Yes, yeah.
Yeah, so the safety is a real concern, right? And so that's why there was so much monitoring. In principle, the intervention should have no impact whatsoever on any human condition. And we, but how, that's lovely to say, but how do you actually do it? So there was a big safety aspect to the trial. There were no safety adverse event signals whatsoever associated with the intervention. And there was, you can imagine from the protocol, a whole raft of things being measured that we thought might capture some negative outcomes. Getting the real issue is not, the WHO normally wants a confirmatory trial, another one, kind of like the FDA requires 2 large-scale randomized clinical trial for regulation or approval of a drug. They don't do that. They suspended that for vaccines. So that was sort of behind the expedited approval for vaccines. But the WHO, we took evidence that was not randomized from other places. So there's a lot of, we have done deployments of Wolbachia elsewhere. In Australia was one where we've collected observational data and we have interrupted time series. There's someone I heard about at the unit who's doing interrupted time series and COVID. Yeah, so I'm curious to see their work, but because I heard it when I was listening this week. So we took all of that evidence and the WHO approved. There's a technical term the WHO uses that gives their stamp of approval that this is an intervention that can be used now. But it is, again, a long political process with the WHO. But I don't know if you have other ideas of how safety could be assess other than monitoring. What happened in Yogyakarta is now, as part of the whole trial, the whole city is now covered with Wolbachia. So that was part of the deal, that if it worked at no cost, we would go in and deploy them citywide. And now every year I'm watching the dengue. It's way down, but dengue goes up and down quite dramatic. That's a sort of cycle of up years and down years.
And anticipated consequences on the mosquito itself, and super mosquito that does some other things.
There is some concern about evolutionary push. What will happen to the wild Aedes aegypti population if they all have Wolbachia? Will dengue mutate in some ways? Because it's now going to live in a world where Wolbachia is in a way it hasn't. And what will happen to the virus? So there are long-term concerns, and there's also an issue if it's really as successful as we think it is. In 10 years, everyone in Yogyakarta will be naive to dengue. And if something happens to the virus that evades the protection, and you put it into a completely naive population, you may have problems. So there are mathematical modelers trying to think about these questions as to what will be the evolutionary impact of that. I think it's less worrying than GM mosquitoes myself, but that's just a personal opinion. But Oxitec has the same issues to worry about with genetically modified mosquitoes.
I think we can, I'm aware we have a drinks reception to get to, so we could maybe take one more question if anybody has one. Sean's shaking his head.
Let's have a drink.
We can continue this conversation over drinks, I think, then, in which case, thanks again to all our speakers, and particularly to Nick. That was fantastic.